Hi everyone. Hi. Welcome on in. How are y'all doing today? Are you in this paper? They changed the scales on a chicken's leg to feathers. Why does this? And they didn't do it by adding genes. They did it by manipulating existing genes, and in particular, members of the sonic hedgehog signaling pathway. What is a signaling pathway? A pathway is one protein or molecule communicates to another, which communicates to another, which communicates to another, and it does something. Sonic Hedgehog, signaling on its own, what it does is it's important in developmental biology. Actually, we're gonna quickly do a little bit of a jump on just what Sonic Hedgehog signaling is. And it's the same reason why you have certain body parts, why we have a certain number of fingers. So it's very critical in development. And I'm just going to give you all the crux of it now of why this isn't... This paper is getting a ton of media attention. And the reason is because it's looking at if... It's analyzing the connection between birds being dinosaurs, right? If you can change scaly bird bits into feathery bird bits... That almost says like how genetically close birds are to dinosaurs, and again kind of solidifies the hypothesis that in terms of an evolutionary lineage, it's very close. Yes, Smikes, please ask away. Your favorite reptiles? Noise. Birds are birds? Does it have something to do with going fast? It does not, RPG fan. Actually, there's a bunch of different hedgehog signaling pathways in developmental biology. And what's wild, RPG, is that they're named different things. There's desert hedgehog. There's a couple of other ones, but the first one was named after the video game character. We'll talk about it. Like, there's a little video of like they're talking about why they named it that. But there's RPG. It's not because of anything speed. It's just because, oh yeah, this is kind of cool. And that's the end of it. It was just like, oh, that's kind of cool. Hi, Grimly. How you doing, Grimly? That is true, Smikes. It is true if we're looking at the evolutionary lineage of there. Insurance disaster reminds me of your baby photo disaster. Yeah, travel the world. We've had some interesting people that we've interacted with, right? Travel. The nice part about it is at least we did get the baby photos. And if that's any indication, maybe we will actually end up getting the downstairs fixed. At least that's what I'm hoping. But so a lot, Smikes, of the popular media have just been like really harping on this paper that this is showing that correct evolutionary connection between birds and dinosaurs. Those popular media articles fail to mention is that there have been papers that showed this in the past. So this is one set of developmental biological genes and pathways, and they're regulating things in a very unique way. But there's also a signaling pathway called Notch that has been shown to do the exact same thing. And because it's been shown to do the exact same thing, like, you would imagine maybe this should not get such a big deal. The only difference between that paper and this one is that in this one, instead of Notch, it's Sonic Hedgehog that they're showing. We're going to talk more about all these things, but a lot of the popular media articles are not citing the previous study, and they're just looking at this one. Cool name self. I think it might be something with like the university contacting folks, RPG. So it's RPG, some of the behind the scenes stuff when your papers get media attention is very strange, where you'll get media attention if the university reaches out. Like, the university has a PR department, right? If they reach out and want to do a press release on your study and how much like they want to push it, that can influence how much like folks are going to pay attention to it. Like, we had press releases on some of our papers, and some we didn't have any press releases. If we didn't have any press release, almost all of those all of those papers, or I should say none of those papers actually got any media attention. If there was a press release, then yes, then they got media attention. So my guess is that something like that happened, but at least the popular science articles should have researched what they were talking about. But let's go ahead and go back in time and actually take a gander at what sonic hedgehog signaling is. We're going to look a little bit into, this is a very silly video. There's Sonic, not Sonic, but Sonic.
Don't you like me a little
I
bit?
think Amy.
Ew, I don't even like you a little bit.
I heard you like me a little bit.
Well, maybe just a little bit.
Guys, yeah, checking out Legendary Big Gaming, game developer extraordinaire, working on the Homeward Hounder, Dog's Life, or I'm a Dog. Also one of the best fathers in the world. Give it up for Bit Gaming, and please, chat, say a little prayer to his computer. Chat. Tell Big Game is amazing. Astronomy show that Pikachu pathway should be next. So the reason Astronomy show it's also named Sonic Hedgehog signaling is because it was discovered in fruit flies first. Fruit flies have no naming convention in terms of what you can name things. So Astronomy show, if you were to identify a new pathway in fruit flies that had not been identified in another organism, and there's nothing similar in there, because there can be overlap in terms of naming. So like my friend Nate and I, we discovered some genes that hadn't been discovered in fruit flies before, but because there was something similar in a different organism, we had to call it that existing name. So it was a glycosylation gene, and it was called, in a different system, it's called MGAT, and we had to call it MGAT as well. So we couldn't change the names, unfortunately. But Astronomy show, if you discovered something brand new,
together we can rule the galaxy,
then it 100% could work. Hi, Cricket! Cricket, how you doing, man? Guys, love that you amazing. Cricket is here. Cricket, I hope you got the sticker and you like the sticker. Hi, Cricket. Using pop culture reference gets your paper seen. No. Usually not. Astronomy show, so like this, the reason why this was seen wasn't because Sonic Hedgehog signaling.
my best friend.
I think this was seen because of a marketing thing, in my opinion. Guys, please go follow lovely and amazing Cricket. She's live right now, so if you give her a follow right now, she'll go. So please go follow Cricket so she can go. The whole thing to do that. So yeah, Astronomy show, I think there's been other papers. So Astronomy show, there are fruit fly papers that demonstrate mutants that die early, and they're named after Kenny from the South Park character Kenny, where they die rapidly. There's another one named after a Star Trek character called Red Shirt. Again, rapid death. Astronomy showed those papers got nowhere, like in terms of visibility. And so it's where they're published, how they're published, where is like really a big factor. And then if there's any kind of snazzy name for it, then maybe there's something there. But if there's nothing snazzy and silly, no, Astronomy show, I remember them because I actually got to work with them a little bit. But beyond that they're silly as well, right? Like they're also a silly name. But I don't think there's, like, outside the scientific field, I don't think there's much of that, if that makes sense. So this is, again, the Sonic Hedgehog signaling pathway that we're talking about. And so we're just going to go a little bit into the pathway. This looks a little bit, again, it's a silly looking image that you see here of Sonic. This is the Sonic hedgehog, as it were. But let's just do a little bit of what the signaling actually like. It's very simplistic on this, but it's demonstrating connections between molecules.
Let us dive into the pathway. So let's just examine it on.
So this is an example of how one of these pathways work. If the membrane of a cell is in this yellow, there's a receptor and it talks to different molecules. And these molecules interact to eventually do something downstream. Meaning at the end of the pathway, something will happen.
Side level without Sonic Hedgehog. So we see there's patched 1, it's a membrane protein. And through a certain mechanism that I will go into later, it is stopping the smoothened protein from entering the cell membrane. And
is the interesting word in the title that is Sonic Hedgehog signaling, which is what we were kind of focusing on, which is a developmental signaling pathway. It's complicated. There's a lot of molecules involved, but I was trying to highlight what a signaling pathway is, where you have, this is the cell membrane, there's receptors that detects outside of the cell, and then things would change. So the receptor that it's detecting a molecule called Sonic Hedgehog, that is SHH, and this is the state of the cell without the Sonic Hedgehog molecule. And then if you add the molecule, it changes.
But inside the cell, a protein known as SUFU is inhibiting a transcription factor known as GLI.
So this SUFU is bound to GLI, and it prevents GLI from binding to DNA. So that's just like how this whole system's working. There's interactors, there's inhibitors. It's just like a giant map of players in a game, almost. A good way to think about.
By binding to it and creating a complex. And in these complexes, GLI is then phosphorylated by kinases.
There is a lot. The reason, so this video is not ideal because there's a lot of terminology that doesn't actually go into explanation. But again, the reason I'm only showing part of it is just to show like some rudimentary what the pathway looks like. A kinase is an activating molecule. Some phosphorylate, meaning they add a phosphate molecule onto something, and that will activate that molecule. So if something is phosphorylated, it's been given a phosphate, and that activates the molecule to do something. And that's the role of a kinase. So again, we're getting into the weeds here, but again, it's just I just wanted to show what the fundamental pathway looked like.
This phosphorylation then marks it to be cleaved by proteasomes. And this cleaved clee is then able to enter into the nucleus.
So by putting the phosphate on, the GLI can go into the nucleus and turn on genetic information.
And block the traditional GLI targets, which are usually growth-related. So as we can see, without the Sonic Hedgehog, which inhibits the patched, the cleaved GLI is able to block such targets. Now, let's examine it a little more closely. So patched 1 inhibits smoothened by way of the regulation that GLI, the nucleus.
And then here now, we're gonna, I'm just fast forwarding. If you give the Sonic Hedgehog molecule,
is free,
then the SUFU now isn't being targeted for destruction.
GLI, as a transcription factor, is now able to do its job and enter into the nuclear membrane and activate its targets.
So because it didn't get hit with that kinase, it didn't get that phosphorylation state, it can now enter the nucleus and turn on key genes. When it is phosphorylated, it still enters the nucleus, so it's still in its active form, but because it's phosphorylated, so it has that phosphate on, it changes shape, and that shape change means it can't correctly do its job on the DNA. So that's some of the fundamental regulation that happens of these downstream molecules. And the only reason this is important is, in terms of the paper, is because they actually, so even in the paper they call it the SHH pathway, so the Sonic Hedgehog signaling, that's that same molecule. They altered some of that signaling with or without inside the legs of chickens to get different results. So very, very rudimentary biology. Let's talk a little bit more about the Sonic Hedgehog gene and what it does in your body, because it is conserved in humans, it's conserved in flies, mammals, tons and tons of creatures.
Today on It's Okay to Be Smart, thumb's the word.
And they're focusing on fingers because the Sonic Hedgehog, because it's involved in development, we do know that different doses of hedgehog signaling contribute to having different numbers of fingers. So polydactyly, which having more fingers than expected, is a sign of changing amount of that Sonic Hedgehog signaling.
The ancient Greeks called it the second hand. It could kill a gladiator or even set off a Shakespearean tragedy. It could build a tool or even... Hold on a second. Now, it's hard to put a finger on the best evolutionary adaptation, but thumbs are up there. And we're not the only animals with opposable digits, but it's hard to imagine being human without those little guys. So what can a little blue hedgehog and an ancient fish teach us about how these guys evolved?
Is that why Tails has two tails of Sonic Hedgehog signaling? It would be why, Smikes, yes. That he had, well, actually, no, I have no idea. Actually, I think Tails is referred to as a he in the game in the shows, but it is voice acted by typically women. So I have no idea now. I think Tails is a boy, though, in the game. Anywho, doesn't matter. Yes, there is a different amount of Sonic Hedgehog signaling in the tail during development, and that's what creates the doublet tail in Sonic. Not Sonic and Tails. Hi, Kai! Welcome in. Kai, I mailed off your sticker today, Kai. Or, yeah, it's your sticker. Sorry, I'm just making sure that I mailed off the right thing. Yes, the sticker. Most people got a magnet this time around, but you did request it. Was that intentional? No. Smikes, the gene was discovered after the video game was made. So the video game was made first, and then the gene was named. So I think in the naming, it's just they liked Sonic the Hedgehog, the video game. There's no cool scientific reason, unfortunately, there, Smikes.
To find out, I hitched a ride to chat with paleontologist Neil Shubin. All right, I'm here with a really special friend today, Dr. Neil Shubin.
Great to be here, thank you.
So you work at a medical school. Why does a medical school need a fish paleontologist?
The reality is often some of the best roadmaps to our own bodies lie in other creatures. An entire branch of the tree of life is embedded in every cell, every gene, every organ of your body. And our challenge as anatomists, as geneticists, is to untangle that history.
Neil studies both the past and the present. By looking at modern genes, he can study how living chemistry...
Fortuitous Charlie alert! Thank you, Fortuitous Traveler. So again, a paleogeneticist is trying to analyze the evolutionary lineage of cells and genes and how they're becoming not just what they are today, but what other information's embedded in that genome. And that's a little bit of what the researchers did in this study.
He molds all those endless forms most beautiful that Darwin talked about. And by digging up ancient fossils, you can see how our bodies have changed over time.
So Tarzan, if you do go to schooling for this, make sure that you get into a good program for it. So you'll need to go to there's probably graduate schools that you'd want to go to, not because of the name of the institution, but because of the researcher whose lab you'd be working in. That's always a difficulty of this, Tarzan, is identifying the right lab to work in and get the training to be the expert in the field and for it. Watch the stream, never watched before, lurking here till Dunsies with the day. And hi, Makuaka. Welcome on in, Ma.
And one of those fossils in particular has completely redefined our journey out of the water.
Okay, so this is a cast of a skull from a creature that's 375 million years old. And it's from the Arctic, about 600 miles from the North Pole. It's a fish, but it's a special kind of fish. It's called Tiktaalik. It has a paired set of nostrils in the front. This is a fish that, if I was to show you its body, clearly has scales on its back and fins with fin webbing. But if you look inside the fins, you find elements that correspond to your upper arm, forearm, even parts of your wrist, inside this fish.
Tiktaalik is one of the most amazing fossil finds of all time. It's a so-called transitional form. It's a fish with legs that helps us connect the dots on the evolutionary tree between fish that swim and animals that walk. By studying those ancient bones, we discovered that the arms that carried that fish out of the water aren't that different from these.
Oh, here's the beautiful thing. Take your arm, okay?
Did you know, John, that Hank Green was diagnosed with cancer today? He got Hodgkin's lymphoma. They came out with a statement. He was supposed to do some presentations and talks, and he had to cancel them because he's undergoing chemo now. This is, I think, his brother. I believe this is the brother of Hank Green. I don't know his name, but yeah, Hank has cancer, Jern. So he's had to cancel a bunch of things that he was supposed to be doing so he can get straight into chemo. So once he's cured, John, I'll bring back the Hank shaking my arms around. But until then, we're gonna pause on the arm shaking until he's clear for the cancer. It sounds like, John, that he's saying it's a very treatable form of cancer, and it's early enough where it hasn't metastasized, but he still has to undergo chemo. So there's never any guarantee, but I'm hoping that everything will be all right. I know, and John, we watched so much of Hank, and I was just like, no, not Hank Green.
You have bones inside here. You have one bone here, two bones here, lots of little bones in through here, and then your fingers, right? When we look at the fossil record, what we can see is the assembly of this one bone, two bone, little bone finger pattern. What Tiktaalik has, it's a fish with a fin. But if you were shed out those fin rays and take off the scales, what you would see are versions of your upper arm, elbow, forearm, and wrist.
So there, how you have the conservation of the same shape as our hand, what the genetic determinant of that is sonic hedgehog signaling the exact same genetic determinant in the fish as it is in our hands.
In this fish. And then we can trace that all the way up from the fish to land-living animals, and we can trace it even back further down from Tiktaalik to fish. That's what we do as evolutionary biologists, is look at the assembly of this beautiful organ here.
Wow, so look at this arm, guys. This is 375 million years in the making. So what does any of this have to do with making thumbs? Well, to get this, you need this. And to get any of this, you have to be born. Now, all of us started as one cell from a sperm and an egg, and that became the trillions more that make our bodies.
Think about it. This begins as a little bud, 4 little buds, boop, 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 boop. 4 little buds pop out of your body as a little embryo. And those buds, they push out of the body, filling with cells inside, and that bud begins to form a paddle. And that paddle is where the hand will appear. So as this paddle appears, cells are dividing inside there, and genes are being turned on and off. And it's the activity of those genes being turned on and off that sort of orchestrates the process of the formation of the different fingers in your hand.
Okay, Neil. It's one thing to make an arm, a hand, or even fingers. Even a frog can do that.
Okay, and I also have a hand at the end of this arm over here. See that? Yeah.
But that doesn't explain my thumb. Why does it look different from my pinky?
One of the reasons is because the genes are being turned on and off in different parts of your developing hand in different ways. One patch of tissue right at this corner, right under where the pinky will form, is really important because a signal is produced in that little patch of tissue that...
So this is similar to what we just watched, this video. So with Sonic Hedgehog signaling and without, remember, so that's what we were looking at in this little diagram. With, without, and with. It's not as simple as presence or absence of that molecule. Instead, it's Sonic Hedgehog, the molecule, it's on a gradient. And that gradient is what is influencing what genetic information is going to be translated from the DNA to RNA to protein. And different amounts of it will create different things, whether it be a full hand, a full finger, different amount, like different finger numbers. It's a very elegant system, but it is more complicated than just on or off.
It migrates across the developing fin bud, and the cells respond to that signal to develop different fingers in different ways.
What's that guy called?
Sonic Hedgehog, after the video game.
After the video game. The fact that I can play video games, hold my controller, is due to
Sonic the Hedgehog.
That's
so Coffee Rocks, it all of a sudden, like, his analogy of like, oh, the reason I can hold the video game controller with my thumbs and play the game is because of the gene. That's not what the researchers, like, if that was the depth that the researchers were thinking, hats off to them, but it was much more I think that they just enjoyed the video game, which is okay. It makes me chuckle though that that's the end result of it, as it were.
Really cool.
Triple cool.
Express too much Sonic Hedgehog in the embryo and you get extra fingers. Too little.
And so this is different in the birds. So in the birds, they changed the level of Sonic Hedgehog in cells in the leg. And these cells in the leg are usually creating this scaly-like appearance, and so they changed the amount of Sonic Hedgehog, and that created feathers. So there it wasn't, depending on where this is expressing, like where that gene is being turned on and off, it'll have different results. It not just is only hands, but it could be expression elsewhere as well. All right, BRB, no worries. Enjoy the video cassettes. Tarzan, enjoy. And interesting that although hedgehog is one word, they break it into two for the acronym Sonic Hedgehog. Bit gamey. There most likely there was already a gene with an SH designation. And that's probably why they changed it to SHH, to be totally honest. I was just trying to think of if I can remember offhand. I remember SHI. That's what I was thinking of, is Shibiri. And that's a mutation that causes neurons to stop firing. We can take a look and see what SH is. Because that would give you the explanation of why that is. Which I will show you how we look this up. So there's a genome browser called Flybase. This is where you can look at any gene, like in the fly genome. And so if we're looking for a gene, let's just look for SH. So SH is the gene shaker. Bit gamey. Encodes the structural alpha subunit of a voltage-gated potassium channel. Plays a key role in maintaining electrical balance in neurons and muscle cells, as well as regulating neurotransmitter release at the synapse. So, most likely, Bit Gamey, Shaker was discovered first, and so it gets the designation SH, and then Sonic Hedgehog will get that SHH designation. The acronym breaking doesn't always make sense, Bit Gamey, but I think it's more some historical elements come into play. So actually, each one of these shorthands, so you see Bit Gamey in the search bar, we typed in SH, and there was this singular gene that popped up. It's because every gene has a designation, and there's a unique one for every single one. Are these universal in species? So the name tactics, this is not the universal nomenclature. The naming scheme, so we'd have to go to... So these are all the ones in humans, for example, tactics. So the human orthologs are, it's Homo sapiens, so it's this HSAP, KCNA1 through 10, CNF1, CND1 through 3, CNQ 1 through 5, CNV. So there's a lot more of these in humans, like predicted, than there are in the one example that there are in the fruit fly. Let me see. Gene model and products, expression data. Similarities. We can see if there's any human disease associated with it. It looks like microcephaly, developmental encephaly, ataxia, heart issues, all can happen with mutations of this gene in humans. And in humans, there's different members of the same gene family, it's called. So tactics, imagine like there's an entire web of similar types of genes that do very similar things. And that makes up a gene family, versus in the fly, for this one gene, there was just one type of this one gene, that was all that was existing. In humans, there's many more, and so mutations in like this KCNA1 leads to certain kinds of ataxia, KCNA2, this epileptic encephalopathy, and so on and so forth. Would that be why some... Did that make sense, by the way, Tactics? Would that be why some birth defects are like shortened, non-functional arms, sonic effect early in development of the arms? So, in part, Coffee, it gets, again, more complicated in humans. There is still sonic hedgehog in humans, but there's also other hedgehog signaling pathways in humans as well, but it operates on a very similar principle. They might have different targets, so it might not be for development in terms of the hands, but it might be development in another part of the body. But it could be a mutation of the actual sonic gene. It could also be a receptor of it, or anything in this pathway. This Coffee is where it gets really hairy for geneticists, because
together Ambrosine! The galaxy.
Ambrosine, thank you for the resub! Ambrosine, 16 months. Thank you so much for that continued support, Ambrosine. How the heck are you doing? Welcome on in, Ambrosine. Thank you so much for that ongoing support. So, Coffee, members of this pathway, and I want to point out that this pathway is abridged. It is far more complicated if you put in all the members. Like, kinases you see as plural. There's a lot of members there. It could be the sonic hedgehog molecule itself that's mutated, and because it's also dependent on a gradient, right? So you can imagine, in Coffee, they've done these experiments in fruit flies. What's happened if you get rid of the gradient? So in flies, you can make a two-headed or two-butted individual, because the embryo is on a gradient of these developmental factors like sonic hedgehog. And if you destroy that gradient so everything is really, really high or really, really low, that gets rid of the gradient, and it gets rid of that directionality of the animal. And so you get double-headed, you get ectopic expression of tissue, which means there are fruit flies where if you turn on and off these genes, you'll get eye tissue all over their body. So it's not like they have eyes, functional eyes all over their body, but there's eye cells on their legs, on their thorax, just growing all over. And so you can do it on the level of the sonic hedgehog, like that molecule, or any one of these pathway members as well, which I think is kind of wild. Hi, Green Mazing, welcome in. Big Gamey says, okay, BSC's bovine spongiform encephalophilia. I'm so Big Gamey. I've been tongue tied now. I've been totally tongue tied, Big Gamey. But yeah, thank you, Big Gamey, for looking at that. I didn't know that, Big Gamey. Hi, Nelf, welcome on in. Encephalitis, thank you, Tactics. I've been reset, Tactics. I've been reset. Just encephalopathy. There we go. I probably didn't get it right, but it's the closest that we're gonna be able to get on that front. But yeah, Coffee, these kinds of pathways are really complicated, not only because there's a lot of members in the pathway, but it makes it even harder that it runs on that gradient system. I hope the baby is growing well. Yep, she is almost actually green. Next week, she'll be 4 months old. She'll be 4 months old next week, Green. Pink encephalant. Oh, encephalanthy. Well, Tactics, I lost it again. Tactics. But Tactics, just so that I remember, I will throw this fly-based report on the mentioned on stream tab. So if y'all wanted to play around with this, again, the whole website is actually pretty straightforward to navigate. You just go to the top of the page and put in a gene. You do kind of have to have an idea of the gene name of what you're wanting to look for. So if we put in SHH, like let's just do Sonic. Hold on. Hold, please. We're We're getting some errors here. Okay, there we go. Aha, okay. So they've actually, so this is a little bit of an update. You see that there's a change in the nomenclature now. So mouse is named SHH. So the mammalian front is Sonic Hedgehog. And then in fruit flies, let's see, it's just hedgehog. That's what it is, because there's only one in the hedgehog. There's only one in the fly, and so that's HH for hedgehog. And then you have in zebrafish, which is this first hit here, so fish. And then the second hit here, which is mouse, has Sonic Hedgehog. I wonder what the A stands for in zebrafish. Let's see. Sonic Hedgehog A. Not clear. This is fly base. There's a fish equivalent to it. Fly base. I have not used it, but tactics. I'll throw this in as well to the mentioned on stream if you wanted to dissect and analyze. But yeah, here's the hedgehog one, and see it's encoding in the hedgehog signaling pathway ligand. We can go and let's see coffee rocks if you're interested in human disease. So these are the three in humans coffee. There's desert hedgehog, Sonic Hedgehog, and Indian hedgehog. And the disease states for desert hedgehog, you have sex reversal and gonad dysgenesis with neuropathy as well. So it looks like neurological failure to develop. For Sonic Hedgehog, developmental issues. The encephaly word that I can no longer say. There looks like there's brain formational issues, holoprosencephaly. Yeah, there's a whole bunch like with your nervous system that also goes wrong. And then with Indian hedgehog signaling, brachydactyly, so like different amounts of fingers and hands, dysplasia. So here are all the disease states of it. So coffee, I'll throw this into the mention on stream as well. And it's a pretty good resource if you're interested in like what other disease states can be caused. You can just look in here and you can click on one of these. Like let's say you're interested in this particular result. And then it brings you to this another page that actually like dissects where the publication is. And it even tells you like this gene the thing that ends up being caused is on this certain chromosomal position, like where it's located in the genome, how it's inherited, what's the gene number. All right, so what are we talking about today? What are we talking about? We are having this very fascinating study where they altered a signaling molecule during development that led to featherization of a chicken in areas where they should not have had feathers. And that is what we were chatting about today. And yes, as Smikes mentioned, don't worry if you didn't win any of the raffles, have no fear. There is the merch store right there. Thank you, Smikes. Love your face, Smikes. Where we were right now was actually dissecting what the heck the Sonic Hedgehog actually mean. It is a signaling molecule in development. When it is altered, it can lead to different fingers. And this is actually for the crew coming in from Danny. Y'all will know about the fish Tiktaalik. That has like the hands and fingers as it are crawling around outside. Like that's a missing evolutionary link even. And that signaling that gave it those fingers, we hypothesize the same genetic signaling that gives us different numbers of digits. Tiktaalik. Mac84, thank you for the follow. And thank you, Big Gaming, for that dad joke. Thank you, Jody Fish. Thank you, Jody Fish. Jody's like, guys, get it together. It's Tiktaalik, not Goofy Goober TikTok.
One of the reasons is because the genes are being turned on and off in different parts of your developing hand in different ways. One patch of tissue right at this corner, right under where the pinky will form, is really important. Because a signal is produced in that little patch of tissue that migrates across the developing fin bud. and the cells respond to that signal to develop different fingers in different ways.
What's that guy called?
Sonic Hedgehog, after the video game.
After the video game.
So the gene was discovered and named after the video game. That is why it's called Sonic Hedgehog. Otherwise, would have been called something totally different. The geneticists who identified the gene were big fans of the video game. And I think that's really cool. That's also why we have this redemption for those coming on in who are new. Of Antonia. In a Sonic the Hedgehog outfit, because again of the developmental genetics that we like to talk about.
The fact that I can play video games, hold my controller, is due to
Sonic the Hedgehog.
That's really cool.
Triple cool.
Express too much Sonic Hedgehog in the embryo and you get extra fingers. Too little and your digits go from 5 to 4.
So this is in terms of digits and finger numbers for people, but there's also other developmental effects that this can have, which is again demonstrated by the study that we're looking at today, which is altering the amount of hedgehog signaling inside of a chicken or some other kind of bird. In this case, it's a chicken, and in particular, the scaly bits of the chicken start forming feathers, and that's all regulated by via developmental pathways and these gene families.
Wait a second. I think I just figured out how the Simpsons have...
So Sonic Hedgehog is the culprit for polydactyly. Yes, Joey. It's also... So in higher order mammals... Hi, JP! Welcome in. The one that JP has arrived. How you doing, JP? In higher order mammals, so it was originally discovered, Jody... Actually, we were actually looking at this just a moment ago. Ooh, let me get the actual gene list for you, Jody. Oh. Hold on, let me grab. So we were actually looking at the genetic information that we have on these animals. And so if we look in fruit flies, it's just called the hedgehog signaling. But in mammals, where there's more of, there's 3 hedgehogs that humans have. There's desert hedgehog, there's Sonic Hedgehog, and then there's Indian hedgehog. And dysregulation in each of these can cause different amounts of, or can cause different disease states as well. So Sundry, it's during development, and it's also regulated on a pathway, meaning there's different densities. So it's not just, hey, we're doing this thing with our genes, like turning it on and off. Instead, it's a gradient that forms, and high amounts of the gene equals one thing and low amounts equals another. And having that gradient gives the developing body information of how and where to develop. Guys, lovely JP is in his house. Make sure to follow JP, Woodcrafter Extraordinaire. We love JP here. Go follow JP. I heard extra digits is hereditary. Does that mean the gene is familial altered? It can, Coffee Rocks, yes. There are instances where it's mutated in utero. So there can be, as you're developing embryo, you can acquire mutations that can also yield this kind of effect. But there are familial ones. And actually, it looks like, at least on this desert hedgehog, is shown to be a sex-linked gene, so that is mutated, it looks like, on the X. And then Sonic Hedgehog and Indian Hedgehog are mutated on the autosomal, on an autosome, sorry, non-sex linked gene. Hi, Odin! Welcome in, Odin. How you doing today, Odin? It's lovely to see you, Odin. I hope you're doing well. Welcome on in. We're chatting about this Sonic Hedgehog paper that came out recently. Not about the video game character, although that is where the naming system is rooted, but rather talking about the genetic signaling of this molecule. Grimly, my friend, sleep well, Grimly. I love your face, and also know that you are breathtaking and handsome and kind. And one day we will meet, and I'll give you the biggest hug that I can muster. With your permission, of course, Grimly. Love you, Grimly.
Evolve. John Saunders discovered that if you transplant a tiny patch of that tissue on the opposite side of the bud, you create a mirror image wing in chickens. And we don't just find sonic hedgehog in humans, we find it in fish, and frogs, and chickens, and mice, and probably even real hedgehogs. And it does similar things in those animals as it does in humans. Of course, genes like sonic hedgehog play in harmony with many other genes. It's just one instrument in that beautiful orchestra of chemical signals. The search for inner fish and inner hedgehog.
Does it have anything to do with the creation of tarsals and metatarsals? So, Hanny, for this particular one, not necessarily. It's on a gradient scheme, so it's not just responsible for bone formation, but it's responsible for just the generic formation of the hand. So there's it's not just bone, it's the entire structure and also separating out. So there are signaling molecules where the bone won't split, and it's because you have different amounts of the sonic hedgehog, and so it'll just be like a thicker mass of bone. It never separated out. So it just depends on where in the pathway you're mutating things. Thank you, Smikes.
Hedgehogs is the story of how genes and fossils, two different branches of science, come together to form the tree of life. Thank you so much for joining me today. I've learned a lot about my inner fish, and I hope that you have too. If you want to learn more, check out Neil's book, Your Inner Fish. It is awesome. There's so many great stories. You really feel like you're in the field with him. And PBS has a special 3-part series premiering Wednesday, April 9th at 10 p.m. Eastern, also called Your Inner Fish. What do we say, Neil?
Stay curious. My inner fish.
Who's got 2 thumbs and wants you to subscribe?
He does. So why was it talking about a fish? It's because the fish like have the same genetic components that also like create those effects, if that makes sense. What about people born with webbing between their fingers? So Twitch Ag, that's also a function of development. So it's because those cells that are connecting between your hands are supposed to undergo apoptosis or cell death. So if they're not undergoing cell death, that is not necessarily a function of hedgehog signaling acting up and being loosey-goosey, but something else in that same pathway. So it's just not killing off the cells appropriately, if that makes sense. Thank you, Smikes. Smikes, thank you. You handsome devil. So that's just the foundational piece of what sonic hedgehog signaling is. We'll go into the pathway that they altered in a moment, but the big thing that I want to show is why they're actually doing this. Similar to bones, it is really cool, right, Hanny? I mean, we all have web feet. We do. So, Dame Karen, if you look at, there's some National Geographic time lapses of human development. I don't want to Google it offhand, Dame Karen, and not pre-screen the video because I don't want there to be anything unsettling that we watch. But your hands, they open up and there's still webbing between everything, and then there's bones that are forming, but there's still just a giant web. And then eventually those cells undergo cell death and apoptosis, and that's what makes us have these kinds of like fingers and digits versus like that webbed feet or webbed hands. Neuralicia, thank you for the lurk, you breathtaking soul, and thank you for the raid again, folks. If you're not checking out Neuralicia, again, please go drop her a follow. Neuroscientist Extraordinaire streams Mondays on Vulgar Science and Fridays with some science news. Go check out the breathtaking Neuralicia, Neuroscientist Extraordinaire. Love you, Neuralicia. Serious question: can you compete? I do not know, Smikes. That is a legitimate a good question. I don't know if it gives you any kind. Number one, I don't know if it gives you an advantage, and that's not me being facetious. I honestly don't know if it does help or not, because it might not help. And then if it is, I don't know. It sounds like something they would do. For the swimming legislation, there's a lot of interesting bans that they lay down. For example, at one point, they used to allow this particular kind of material for swimsuits. And then in 2008, there was a bunch of records that were being broken at the Olympics, and it was all by people who were wearing that suit. And so what they ended up doing was they banned the suit, and you were no longer allowed to swim in that kind of swimsuit. And so that is the swimming legislation on that front was a little bit weird, Mike. So I don't know. It would not surprise me if they had a ruling on what you could actually, if you could compete or not, just because they have so much regulation on what you can wear. But actually, Dame Karen, hilariously enough, it does not have specific rules. Okay. Well, that's the International Olympic Committee, but I wonder if domestic ones do have, like, to what layer. Dame Karen, my sister actually got a suit like that right after the Olympics. We got one, like a used suit off of eBay, and she was gonna, because the end of season swim meets, she was gonna use it for, but the Olympics finished, and then there was a 2-week block where before that swim meet, and that's when they banned that swimsuit. They were Australians breaking records, weren't they? No, it was everyone. It was not a nationality that was doing better over anything else. There was Americans, there were a lot of people breaking records, and it left, right, and center, and it was everyone who was wearing that suit. And that was the big suit to be worn by that. If you wanted to win, you were wearing that suit. It had nothing to do with nationality. Okay, so let's jump into, again, the paper itself is looking at this phenomena. So the transition between scale growth, and so they actually did a bunch of experiments as well. Phelps was also around at that point. Your team would have been, sure, but so were Americans, Dame Karen. That is not an Australian specific ruling. It would have been Murph. Yeah, Phelps was still there. I think Natalie Coughlin was still swimming. There were a bunch of people who wore it. Phelps would have won without them. There was, I think that was the Olympics where he won the record number of gold medals in an Olympics. And there was one event in the butterfly where it was a hair of a difference. And actually the suspected reason why he won is because he actually did a not incorrect stroke in terms of by law, but by form. So your butterfly is supposed to like go all the way out like this, but he did an accelerated one where he finished like this, and it was a half second like between him and the second place guy, and the reason that little extra oomph allowed him to get a little bit of an extra acceleration in beforehand. And you guys might be like, Blunt, why are you talking about swimming? What do you know about swimming? I qualified for the Olympic trials in breaststroke. I was state champion in 200 breaststroke down in Georgia. I was our high school swim team's captain for both years that I could be swimming since I was aged 5. The only reason I didn't go is because I blew out both my shoulders twice. I was also a swim coach for 10 years. So that's how I know all this, like, background physics and how, like, correct stroke and all. I even took courses and all this. So, like, that's where my background is coming from. I'm just, like, loosey-goosey about it. It was absolutely, Smikes, absolutely. Could you kill off the webbing after birth? So you could not, Coffee. The reason is the thickness of the cells. So I think there are surgeries that you can do, but right now, Coffee Rocks, if you had the webbing and you wanted to induce cell death, the problem is we don't know how to selectively induce cell death in just a small subpopulation of cells, which is what you'd have to do, right? Because you'd only want it to be in between the web instead of everywhere else. And that's actually, Coffee Rocks, that research is also looking to influence answers for cancer, because you can imagine if you have a tumor in your body, if you can induce cell death in those cells but not the cells surrounding it, right? That would be like that's the slam dunk right there. So the similar principle is also applying in that skin removal if you were to do it on the same molecular level, like that induce that cell death. But what you can't do is you can't do it selectively enough, right? So we can induce cell death in cells, but how do you stop it and how do you target it to a particular cell population? And so while you would think that tech should be there, given that's what happens in biology, that level of regulation we just are not familiar with yet enough. Like in development, when you're developing as an embryo, there's a lot of signaling factors at play, not just the presence or absence of signaling factors, but also gradients of them. So concentration differentials and of a lot of different molecules, and all those things feed into that programmed cell death and targeting cells for certain kinds of development. And so you can imagine without all those extra signaling molecules, it's like, well, how do we know what to actually target? And so that's why at Coffee we're unable to do so. But again, I see no reason why eventually it can't be utilized, because I know that there are active research to be able to do what you're asking about, but in the context of cancer. And again, as soon as you crack open the mystery of how do you target particular cells and be able to turn it off, then you should be able to do it in another piece of technology as well. Does that answer your question, Coffee? Not only a great mind, but a great athlete, huge amount of dedication to get to the trials. Oh, it's an insane amount of it. No, if it's to get into the trials, it's like I know we were doing 6 days a week of practice, and most of those days were doubles, and some of those days, in addition to the double practices, also we had what did we call it? Dry land, we called it now, which meant we were doing workouts in the parking lot before we were getting into the pool, like on land. And so like it was, push-ups and like some weight training and like all that jazz as well to be like ready, set, go. Again, it did blow up my shoulder, both shoulders twice, but I still look back fondly at those memories. I don't miss the getting up super early. Well, I get up now super early because of work and the little one, but getting up super early still wasn't my thing then. What are the scales? Are they just weird skin, or are they like hair or feathers? So, Hanny, these are scales on the chickens, like on just their legs, like the scaly bits on the chicken's leg. It's like keratin that it's based off of. It's just like thick layers of keratin. And if you change the signaling in there, you can cause there to be feather growth. So this particular one is that they treated the cells like during development with 2 different compounds. DMSO is the agent that you do for your control. So it's if you're injecting a group of cells, there's usually an agent that you're injecting your thing with. So like your genetic construct that you're trying to turn off something in the pathway has to be dissolved in something and delivered in a liquid of some kind. DMSO is a very good dissolver, and so that is usually then your control that you're comparing it to. So you have one injection protocol where you put everything into it but the genetic tool that you're throwing in, and then your second one is your experimental. You put the same liquids in plus your extra genetic factor that you're using to modify the genome in some way, and those are the 2 that you're comparing. So here it's the DMSO control and then this SAG treated, which is altering the SAG gene in the sonic hedgehog signaling pathway. And so alter it, so and they took images as well of like dorsal, ventral, and lateral, and to have some comparisons. So this is a control gene that you're knocking out, and then here is a sonic hedgehog that's also changed. And you can see these are the controls, and you're seeing what normally things look like, and then you're getting these feathers in the ones that remove the sonic hedgehog, which is remarkable. The reason we're actually talking so much about this paper is because there is a lot of media reporting on this publication that it's a little bit blown out of proportion, to put it the best possible way. So what it was supposed to be was this paper actually builds on existing work. There are other genes that you can do this with beyond sonic hedgehog signaling. There's also notch signaling. Notch Coffee, you asked about this earlier, is an even a higher regulatory agent, so it's even a higher in terms of the genome regulation in the pathway. And so if you turn off Notch, you're dead. That's how important Notch is. But you can alter members of that pathway because it affects so many different things that it also yielded feathers on this kind of particular site. The popular media articles, though, are reporting this paper as like kind of the first of its kind, and it's, you know, it's disappointing, so I wanted to highlight that it wasn't the first one. Were these flight feathers or feathers for warmth? So Henny, they don't actually hypothesize what, because these feathers aren't naturally in the birds, right? Like they're not having feathery feet. So they're just altering gene expression to get feathers where there were these scales on the feet of the chicken. What they're utilized for, we're not sure. Like what in terms of functionality these are actually involved in. They didn't go into it. They didn't even hypothesize really. It was much more about the genetic pathways and the evolutionary lineage of what this implies versus the functional significance of it. Did they change the gene in utero in smallest number of cells to get this effect on the whole bird? Yes, so Coffee, they did this in a select group of cells. They did not do it in all the cells of the organism. They only did it in cells that are going to be expressed in leg development. And you might be like, how the heck do you do that? These cells at birth or at the inception of the embryo are no different than the cells that are going to create the heart, the liver, the brain, blah, 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 blah. That's because also on all your cells, you have the DNA to create every one of your pieces of tissue in your cells, right, Coffee? But because for some reason, right, some cells became your leg cells. And that's because of that developmental signaling and the gradients that are established. So and there's also genes that are like leg-specific genes, and they're turned on because of certain signaling molecules. So if you alter a leg-specific gene, so it's called a promoter gene, Coffee Rocks. A promoter gene means this genetic element ensures that this particular gene is expressed in this particular tissue. So you put a promoter element on your mutated sonic hedgehog, meaning that this mutant sonic hedgehog is only going to be expressed in the tissue that you want it to be expressed in. So if you did a whole body mutant, it's probably going to be lethal, because there's a lot of signaling that you can't mess up and get away with. But let's say it didn't. You might get something where you're getting feathers in the place of the eye or elsewhere. So this is why you attach it to a promoter. So the promoter, that leg promoter, for example, Coffee, is not turned on and not utilized until after those legs have already started to form. So like, yes, after the leg buds, but before you're forming the actual scales on the leg. So it's not that you form the scales and then from the scales grow the feathers. It's that when it would have actually done the formation of the scales, instead of that, because you've altered that genetic element, you now get the feathers. Does that make sense, Coffee Rocks? It's Mike's. I do not remember, good sir, my 200-meter breaststroke. That was my best event. That was the Olympic qualifying time, but I do not remember what it was. It was, I have a vague memory it was 2 something, but I have no idea of 2 what. Guys, we also have a couple of other friends in his house. Make sure to follow Carlo, the Stream Scribe. He joins us on Mondays for Scriboscopy Monday. Live microscopy with live letter art. Go check out the lovely, amazing Carlo, a.k.a. the Stream Scribe. Love you, Carlo. Love you, Carlo. So does that mean the scales are feathers, but in a different form? They are... It doesn't necessarily... That's a hard question, Murph. It means that the genetic elements are in the cells that could create both feathers or scales. It doesn't necessarily mean, though, that they're the same thing. It just means that the genes that are responsible for creating those same things exist in the same cell. But it would not make them the same thing. Because, again, it's while it might be the same pathway, the pathway is behaving differently. So different amounts of sonic hedgehog, turning it down creates the feathers, turning it higher creates the scales. Thank you for asking the questions, Coffee Rocks. Coffee Rocks, you make it fun and we have a good time. Coffee Rocks. Because of people like you. Does that make sense, Murph? It's kind of a double-edged question that I want to answer like as best I can, but it gets into some hairy territories as well. Yeah, but great question. Absolutely great question. So dinosaurs likely had both scales and feathers that were made by the same. So Mirth, one of the arguments that they put forth is in dinosaur development, because again, the hypothesis is that the evolutionary connection between birds and dinosaurs, that birds evolved from dinosaurs. The idea is that there might have been the same effect, but through different amounts of sonic hedgehog signaling throughout the body of these animals. And that's actually even illustrated, Murph, inside the chicken, right? Because there's parts of the chicken that have feathers, and there's parts of the chicken that have these scaly-like components. And there are also hypotheses, Murph, that some dinosaurs as adolescents, right, that they had feathers and then lost those feathers as they developed and aged. And so that could also be a component as well. Like maybe it's some, depending on the dose of sonic hedgehog, you're getting different feathers with different properties, and that might be a function of that. Does that make sense, Murph? Hi, Daybreak. We're talking about this new paper about sonic hedgehog signaling and its connection to feather growth from scales of chickens and how that connects to possible dinosaurs, because that's the whole thing. A sonic hedgehog in an elevator pitch, it is a signaling pathway involved in development. So the signaling pathway, when messed up or perturbed in some way, will mess up development. And it acts on a gradient system where it's not just an on-off, the gene is off, the gene is on, you get one or the other. It is a gradient in the body as the organism develops, leading to certain developmental effects. And Murph, in humans, there not just is one hedgehog signaling gene, but there's three. There's desert hedgehog, there's sonic hedgehog, and there's Indian hedgehog. And they're all involved in signaling. And when you turn them off, you get disease states. Well, activation potential is more of like on a hill, right? It's instead on a gradient system. A potential energy, you have to get a certain threshold before you have anything happen. Here, it's the entire gradient can have stuff happen. It's just the differing effects. How would things like sonic hedgehog develop? Would it have to have been created before land animals hit? So no, Hanny, it looks like in Tiktaalik, the missing link from a fish that also lived on land, they had activation of sonic hedgehog. It looks like that's what yield. We know many animals have developmental components dependent on sonic hedgehog signaling. We know fruit flies have it. So you're talking in the insect order has it millions and billions of years ago. And so most likely, given how much that gene is conserved across the tree of life, it's probably quite an ancient thing. How would it have developed is probably a need for having, as you, an organism, are developing and you're having features that, I think probably the hypothesis, Hanny, is the most ancestral state is having the gradient. So if you're having a head and a butt form, the easiest way to do that is to have a signaling gradient. So you have a high concentration of a molecule that tells the body that this is the head, and a low concentration of the molecule tells you that this is the butt. And that is the feature that sonic hedgehog works on, but like on a more local level, for example, your hands. So most likely that, you have the global organismal level evolve first, and then you can have it be, evolved for these smaller, more distinct features. Does that make sense, Hanny? Tell me, Hanny, if that makes sense. I hope it makes sense. Guys, go check out the lovely and amazing Daybreak Stations, aka Daybreak Studios. Love you, Daybreak. Excellent, thank you. And Hanny, it is so great to see you. I haven't seen you in a little while, Hanny. It's very good to see you. Okay, so how does this all connect to dinosaurs? This is one of, if you know dinosaurs, which you all know if you're from Professor Dan Anduza's stream, Dr. Jack Horner talks about building a dinosaur from a chicken, and I thought this is a nice connection piece to the science that is being done in this paper as well. And because they're actually trying to do this kind of the science on, like, in their lab. So this wasn't, the paper that we're talking about isn't from the Horner lab, but it's a similar biological concept that they're trying to study as well. Dino chicken indeed. No, you, Daybreak. Not the handsome man part, but the sneaky and the naughty part, because you know that always happens, Daybreak. How dare you question it, Daybreak? Is it a dinosaur or just a chicken that looks like a dinosaur? It is a chicken that looks like a dinosaur, Smikes. It is trying to identify the genetic factors that gave dino-like features originated from birds, because as Smikes will tell you, we cannot get DNA from fossils. Actually, we'll look at that after this. Yeah, that's a good one. Well, of why we have to do it like this versus another way.
When I was growing up in Montana, I had two dreams. I wanted to be a paleontologist, a dinosaur paleontologist.
I'm not familiar with Dawes Law, Murph. Tell me about this Dawes Law. If only we could find a frozen dino, Sundry, even if we did, it would be no good. DNA breaks down after 1.2 million years at the longest. So even if you found a frozen dino, you're not going to have something else in there. You're not going to have something else in there. I don't even know Dolo's Law, Murph. I do not know Dolo's Law either. And yeah, the RNA is the precursor. Sundry breaks down if you look at it the wrong way. Like in the lab, Sundry, we have to use this thing called RNase ZAP. RNase is the molecule that eats up RNA. So any molecule that ends in ase eats up the thing that it's named after. So there's DNase that eats up DNA. There's proteinase that eats up protein. RNase eats up RNA. So we use RNase ZAP to get rid of the RNase. Like on our desk, there's on your desk right now, on your skin, Sundry, there's RNase. Why? Because it eats up potential viral elements, because they are RNA based, are viruses. So it's a defense mechanism. So if you can wipe down your desk and get rid of them, but in about 15 minutes there's more. And so you can't do RNA sequencing unless you are in a very sterile area. So that's why you can imagine they're not going to last long out in the wild. It states that once a complex biological trait is lost in a lineage, it cannot be regained or re-evolved. So no, Murph, it doesn't violate the law, because you're not doing anything. You're not evolving the animal, right? There's no evolution that's happening. All you're doing is modulating the genome by hand. You're not inducing evolution. You're making a genetically modified organism, and that's like you're trying to assess what the traits are. And so you're seeing what the genes are and how to alter them and what to do with the alterations to make a certain kind of feature on an organism. And so that's, again, number one, it's not acting through evolution, it's acting through us modifying things. And then again, it's the genetic information's there, it's how the genes are expressed. And it could be too that maybe that gene that originally was for a trait X maybe was duplicated or mutated and led to now trait Y. And if you remutate it back, then it would be able to have those features again. But birds separated. Well, what about Mirth? That doesn't matter, right? Because if there's any genetic information still left over, you can still pull them back to that original state. And again, you're not evolving them back, you're just altering gene expression during development. In us, Murph, we have tails during development. Can you make a person a fish? You can. During development, you as an embryo are very close to a fish embryo. You can breathe in liquid. You have gills during development. As Sundry points out, some humans are born with those partial tails, and then there's no human born with their gills. But those gills are absorbed and you have formed lungs. So yes, you 100% can. You have the genetic information. Our genomes are 70% similar, as Mike says, to fruit flies. It's just like 99.9% similar to bonobos. Why don't I look like a bonobo? It's because of how the genes are turned on and off during development. And that's what we're getting at here. And so that's what this study is trying to look at, of Jack Horner is trying to alter the effects of a chicken and trying to revert them to a state of dinosaurs. And again, it's the same principle as like when we're trying to clone the woolly mammoth. You're not actually cloning the woolly mammoth. You're changing some of the genetic information to make it more woolly mammoth, an Asian elephant more woolly mammoth-like. Same thing as your feature selecting here in this study to create a bird with dino-like features. You're making a GMO. Everything's GMO though, Murph. Everything is genetically modified in some way, whether it be directly through pipettes or whether it be us actively selecting for a trait. It's all going to be some kind of like that.
And I wanted to have a pet dinosaur. And so that's what I've been striving for all of my life. I was very fortunate.
Right, Murph. So selecting for features like farming, even if you're not changing a feature with a pipette, you're still selecting for a feature. And therefore, even if there's been no pipette in a tomato, it's still genetically modified because it was selected for a given trait. Hi, Chris TS. Chris TS, are you feeling better, Chris TS? How's everything going? Has the rain stopped? Are you doing well, Chris TS? Orange carrot is a GMO of a natural black, exactly, but not GMO with pipettes. It's through us selecting for it. Chris, how are you doing today? Tarzan, welcome back, Tarzan. What have I missed? You gotta check out the VOD, Tarzan. I do not remember when you went to lurk mode. Tarzan, Chris TS still in warning state. Be safe, Chris. Be safe. I worry, Chris TS. I worry.
Early in my career, I was fortunate in finding things. I wasn't very good at reading things. In fact, I don't read much of anything. I am extremely dyslexic, and so reading is the hardest thing I do. But instead, I go out and I find things. And I just pick things up.
No worries, Tarzan. Luckily, Tarzan, this is a great time for me to plug the YouTube. If you missed anything, you can check out the YouTube page. Subscribe to the YouTube page. It has all the extra content on there. Trimmed down the science stream. And then also there's also the VODs, but you go check out the YouTube if you want to. If you missed anything that you thought was cool, you might be interested in, and also all the data that we talk about is on the Discord on the mentioned on stream category. So go check that out as well.
I basically, practice for finding money on the street. And I wander about the hills, and I have found a few things, and I've been fortunate enough to find things like the first eggs in the Western Hemisphere, and the first baby dinosaurs in nests, and the first dinosaur embryos, and massive accumulations of bones. And it happened to be at a time when people were just starting to begin to realize that dinosaurs weren't the big, stupid, green reptiles that people had thought for so many years, and people were starting to get an idea that dinosaurs were special. And so at that time, I was able to make some interesting hypotheses along with my colleagues. We were able to actually say that dinosaurs, based on the evidence we had, that dinosaurs built nests and lived in colonies and cared for their young, brought food to their babies, and traveled in gigantic herds. So it was pretty interesting stuff. I have gone on to find more things and discover that dinosaurs really were very social. We have found a lot of evidence that dinosaurs changed from when they were juveniles to when they were adults. The appearance of them would have been different, which it is in all social animals. In social groups of animals, the juveniles always look different than the adults. The adults can recognize the juveniles can recognize the adults. And so we're making a better picture of what a dinosaur looked like, and they didn't just all chase jeeps around. But it is that so.
Hi, Gwendolyn P. Hi, NMD. I saw you guys. Go check out the amazing NMD.
My name is Gwen Stacy. I was bitten by a radioactive spider.
Is this topic number 10? Gwen, is it even topic number 2?
Ahoy there, feckin' scientists!
No, Gwen, it's still topic number 1. Hi, Gwen! Hi, Gwen. How you doing, Gwen? Great to see you. Hi, great to see you too, NMD. Malassi, how you doing today, y'all? Welcome on in. Yes, Gwen, it's a really cool paper. It has a lot of different features. It's talking about both sonic hedgehog signaling. So we talk about a lot of developmental signaling, like the developmental pathway, not just in birds, but how they relate to mammals as well as insects. And then we're correlating it back to like a big part of the study, which is analyzing, you know, how this drives into the evolutionary relatedness of things like the birds and the dinosaurs and the similarities between them. Yes. So, and of the paper I'll post on the Discord, Gwen. What's nice is it is an open access paper, and they actually released a video as well from that lab of like how they did the injections of actually modifying the embryos of the chicken. They actually released this extra YouTube video the day of the paper being released. And the only reason I came across the video, Gwen, is because I followed the author on Twitter. And he posted about the paper, and they're just like, here's this video of, like, the thing that we're doing, blah, blah, blah. And I was like, oh, that's kind of cool. So we'll watch that as well, too, Gwendolyn.
Social thing that I guess attracted Michael Crichton in his book. He talked about the social animals, and then Steven Spielberg, of course, depicts these dinosaurs as being very social creatures. The theme of this story is building a dinosaur, and so we come to that part of Jurassic Park. This is, you know, Michael Crichton really was one of the first people to talk about bringing dinosaurs back to life. You all know the story, right? I mean, I assume everyone here has seen Jurassic Park. If you want to make a dinosaur, you go out, you find yourself a piece of petrified tree sap, otherwise known as amber, that has some blood-sucking insects in it. Good ones, and you get your insect and you drill into it and you suck out some DNA, because obviously all insects that suck blood in those days suck dinosaur DNA out, and you take your...
I actually do like how he's approaching, because we do get that question a lot of can we clone dinos, which is kind of neat. Imagine the Brachiosaurus was a carnivore. The first Jurassic Park would have been horrified. Yeah, so Tarzan, there are interesting hypotheses, right, about like sizes and shapes of these animals and like how that influences behavior. But yeah, what frustrates to me is, and you never can fix it, is you can't ever test these questions, unfortunately, right?
DNA back to the laboratory and you clone it, and I guess you inject it into maybe an ostrich egg or something like that, and then you wait, and lo and behold, out pops a little baby dinosaur. And everybody's happy about that.
Indeed, astronomy show.
And they're happy over and over again. They keep doing it. They just keep making these things, and then the dinosaurs, being social, act out their socialness, and they get together and they conspire. And of course, that's what makes Steven Spielberg's movie, conspiring dinosaurs, chasing people around. So I assume everybody knows that if you actually had a piece of amber and it had an insect in it, and you drilled into it and you got something out of that insect and you cloned it and you did it over and over and over again, you'd have a room full of mosquitoes, right? Or maybe, and probably a whole bunch of trees as well. Now, if you want dinosaur DNA, I say, go to the dinosaur. So that's what we've done. Back in 1993, when the movie came out, we actually had a grant from the National Science Foundation to attempt to extract DNA from a dinosaur. And we chose the dinosaur on the left, a Tyrannosaurus rex, which was a very nice specimen. And one of my former doctoral students, Dr. Mary Schweitzer, actually had the background to do this sort of thing, and so she looked into the bone of this T-Rex, one of the thigh bones, and she actually found some very interesting structures in there. They found these red circular looking objects, and they looked for all the world like red blood cells. And they're in what appear to be the blood channels that go through the bone. And so she thought, well, what the heck? So she sampled some material out of it. And it wasn't DNA. She didn't find DNA. But she did find heme, which is the biological foundation of hemoglobin. And that was really cool.
So it is just iron, which will not disintegrate. You can have that leftover mineral that they're detecting.
And that was interesting. And that was, here we have 65 million year old heme. Well, we tried and we couldn't really get anything else out of it. So a few years went by, and then we started the Hell Creek Project. And the Hell Creek Project was this massive undertaking to get as many dinosaurs as we could possibly find, and hopefully find some dinosaurs that had more material in them. And out in eastern Montana, there's a lot of space, a lot of badlands, and not very many people, and so you can go out there and find a lot of stuff. And we did find a lot of stuff. We found a lot of tyrannosaurs, but we found one special tyrannosaur, and we called it B-Rex. And B-Rex was found under 1,000 cubic yards of rock. It wasn't a very complete T-Rex, and it wasn't a very big T-Rex.
Me too, Tarzan. I always wanted to dig for dinos.
But it was a very special B-Rex. I and my colleagues cut into it, and we were able to determine by looking at lines of arrested growth, some lines in it, that B-Rex had died at the age of 16. We don't really know how long dinosaurs lived because we haven't found the oldest one yet. But this one died at the age of 16. We gave samples to Mary Schweitzer, and she was actually able to determine that B-Rex was a female based on medullary tissue found on the inside of the bone. Medullary tissue is the calcium buildup, the calcium storage basically when an animal is pregnant, when a bird is pregnant. So here was a character that linked birds and dinosaurs. But Mary went further. She took the bone and she dumped it into acid. Now, we all know that bones are fossilized. And so if you dump it into acid, there shouldn't be anything left. But there was something left. There were blood vessels left. There were flexible, clear blood vessels. And so here was the first soft tissues from a dinosaur.
Is dino years similar to dog years? We don't know, Tarzan, because we don't know how long, like how he was saying what the oldest one is, because he was being a little bit silly, but there's no way of identifying what the oldest one is, right? Because every time that you end up digging one up, there could be one older, you know, that you need to dig a little deeper for, right? So we'll never actually really know how old is the oldest that can be. So we don't know about like the accelerated growth rate or anything like that on the front. We can do averages, but until we have a range of ages, we won't be able to figure that out. Which is so it's a hard question. It's a hard question. We don't know yet. Didn't suffer a fossilized death. Yep, exactly. That's on top of it too, Sundry. It's presumably there's a bunch of ones that we'll never capture in terms of ages because they didn't. We won't dig them up.
It was extraordinary. But she also found osteocytes, which are the cells that laid down the bones. And try, we could not find DNA, but she did find evidence of proteins. But we thought maybe that the material was breaking down after it was coming out of the ground. We thought maybe it was deteriorating very fast. And so we built a laboratory in the back of an 18-wheeler trailer. We actually took the laboratory to the field where we could get better samples. And we did. We got better material. The cells looked better. The vessels looked better. Found the protein collagen. I mean, it was...
Sundry! Thank you for the follow, Sundry. Welcome to our colony. Welcome on in. You're one of us now. You're one of us now, Sundry. Welcome.
It's wonderful stuff, but it's not dinosaur DNA. So we have discovered that dinosaur DNA and all DNA just breaks down too fast. We're just not going to be able to do what they did in Jurassic Park. We're not going to be able to make a dinosaur based on a dinosaur. But
Sundry, you're right. One of us. Problem used with this, the acid used with this. Actually, that is not true, Murph. It is not true. The acid is particularly chosen to not destroy the genetic material. So that is not true. Indeed, fortuitous traveler, one of us.
Birds are dinosaurs. Birds are living dinosaurs. We actually classify them as dinosaurs. We now call them non-avian dinosaurs and avian dinosaurs. So the non-avian dinosaurs are the big clunky ones that went extinct. Avian dinosaurs are our modern birds. So we don't have to make a dinosaur. So I already have them. I know you're as bad as the sixth graders, right? The sixth graders look at it and they say, no. You can call it a dinosaur, but look at the velociraptor. The velociraptor is cool. The chicken is not. So this is our problem, as you can imagine. The chicken is a dinosaur. I mean, it really is. I mean, you can't argue with it because we're the classifiers and we've classified it that way. But the sixth graders demand it. Fix the chicken. So that's what I'm here to tell you about, how we're going to fix a chicken. So we have a number of ways that we actually can fix the chicken. Because evolution works, we actually have some evolutionary tools. We'll call them biological modification tools. We have selection. And we know selection works, right? I mean, we started out with a wolf-like creature, and we end up with a Maltese.
So he's just going under fundamental biological selection, and then we could do genetic manipulation to take a chicken which has the genetic information to make something more dinosaur-like, and to just do that. And it's actually, you might be like, well, why are we doing this? Like, what's the point? It's figuring out and identifying how evolution may work, but it's also genetic, developmental genetics, like how genes are controlled during development and what controls them, and how those then correspond to these kinds of features.
I mean, that's definitely genetic modification, or any of the other funny-looking little dogs. We also have transgenesis. Transgenesis is really cool too. That's where you take a gene out of one animal and stick it in another one. That's how that's how people made glowfish. You take a glow gene out of a coral or a jellyfish, and you stick it in a zebrafish, and they glow. And that's pretty cool. And they obviously make a lot of money off of them. And now they're making glow rabbits and glow all sorts of things. I guess.
Fortuitous, who stole your pants?
Your pants around your pants.
Smikes, I was just going to click the sound alert as well. Well done, Smikes. You read my mind. Getting in there. Stuck glow genes in cat. They did, Sundry. Rabbits were very prominent, but also, Sundry, those glow genes are actually a very common tool that we have in the lab. I had fruit flies that glowed left, right, and center in whole body all the way down to one particular neuron and everything in between. It's a great way to identify things, to look at how neurons are connected or certain cellular features are connected within an animal, and how that relates to many, many different factors of the animal. Yes, chat, make sure to follow Irish John Games. Not only is he handsome, but he's also following Gwendolyn P. in being breathtaking. Like my jeans crisper when they come out of the dryer. Yes, Fortuitous. I actually put stuff in the dryer for little baby alone, and I put it on extra dry, but I put it on low heat. Put it on low heat so that it doesn't shrink or close, fortuitous traveler, because that would be terrible. That would be very terrible. I would get the weenus from that. It would be crazy.
Yes, we could make a glow chicken. But I don't think that'll satisfy the 6th graders either. But there's another thing, and there's what we call atavism activation. And atavism activation is basically, an atavism is an ancestral characteristic. Now, you've heard that occasionally children are...
Yeah, astronomy show, it's weird. You can buy kits that you can set up in your garage to do CRISPR, and it's set up in a way where you can't genetically modify anything for what you want to genetically modify. But it's a little kit that's like, okay, you can all of a sudden put this gene into this organism, and it's set up in a way so that you can do like glowing animals, like usually like something small like a worm or a fly. Know someone who successfully stuck glow genes in an ant for a class project. Nice, Murgle. Very cool. Yeah, it's becoming easier and easier to do, where you can even do it with kits that you order online, and like in your garage you can do it. You do need the glow genes. The biggest difficulty, Murgle, is getting something where it makes a genetic line, meaning that you have the organism that you can actually breed, and that involves an injection site, in like where to inject and when in development to inject, because you want to make sure you hit the reproductive organs of that animal to create the next generation where there are all mutants. And that's the difficulty. Making one animal glow is one thing, but having a genetic line of that's much harder to do. Glow genes are the first of all multicellular organisms, the way they work in the last evolved animals too. It's very interesting. As long as it has sundry this UAS side, this upstream activation sequence, and it has something to bind to it to turn it on, you can 100% do it. You can even, sundry, what you can do is take the activation site and an activator that binds to it to turn on the gene from a different animal and put it into something else. So you can do that in fruit flies and yeast. So you take the yeast, UAS, and GAL4 components. The UAS you put in front of a genetic sequence of whatever you want. And then what happens is the GAL4 is a transcription factor. It binds to the UAS and then turns on whatever gene is attached to the UAS. It doesn't matter where that gene from the UAS comes from. It could be from human. It could be from a leopard. It doesn't matter. It's inside the fly. As long as you have that GAL4 from yeast binds to the UAS from yeast, it'll turn on whatever it's attached to. So you can get a little bit of extra modification on there. Hi, Freshie. How you doing, Freshie? Long time no see, Freshie. It's good to see you, Freshie. Welcome in.
Born with tails. And it's because it's an ancestral characteristic. And so there are a number of atavisms that can happen. Snakes are occasionally born with legs. And here's an example. This is a chicken with teeth. A fellow by the name of Matthew Harris at the University of Wisconsin in Madison actually figured out a way to stimulate the gene of teeth for teeth, and so was able to actually turn the tooth gene on and produce teeth in chickens.
What's cool about that too is they didn't put in the gene for teeth. They just took an existing gene that was already there and changed its developmental regulation to turn it on, and that allowed there to be an expression of the teeth inside the beak of the chicken. So that's a very unique way of going about it, right? You're not adding in the gene to get a feature, you're just changing the regulation of existing genetic material. I think it's really cool.
Now that's a good characteristic. We can save that one, all right? We know we can use that. We can make a chicken.
Hi, Ren and Lucy. Welcome in, Ren and Lucy. How you doing, Ren and Lucy? So can we turn genes on vestigial things to work again, like our third eyelid? Unfortunately, Smikes. So in theory, yes. The issue is that it's probably not just one gene. It's probably a group of genes, because you'd not only have to have that eyelid form, but you'd have to have the neurons connected to it so you can control it. You'd also have to have the muscle genes connected to it. So it becomes a lot more complicated to do those features that depend on a lot of different moving parts. So it's not just a single gene, it's a bunch of different things. And so you'd have to identify individual components to turn on and off, and that's where things get a little tricky. Yeah, but in theory, yes. In practice, would you want to? I think the justification would be it would be an amazing feat to be able to figure out all those different genes, how to turn them on and off, and when to turn them on and off. So again, you'd be getting a huge look into developmental biology, which would be cool. The control group, you have to control the other groups with subtle quirks. Well, for the control group fortuitous traveler, you just want those genes in the off state, or if you're injecting something, you'd inject the exact same things, but instead of the genetic altering material, you'd remove that one component from your cocktail, but you'd inject everything else, and that would be a good control to have.
With teeth. That's getting closer. That's better than a glowing chicken. A friend of mine, a colleague of mine, Dr. Hans Larson at McGill University, is actually looking at atavisms, and he's looking...
And this one's the weird one to me, where they are able to get a chicken in development of the egg to grow a tail, but it dies. And they just, last I checked, they haven't been able to figure out what causes embryonic lethality of growing a chicken tail, which is really cool. But of no, it's not dying of embarrassment. No. Is it true we have 180 vestigial organs in the human body? Tarzan, I am not sure how many we have. I am not a human biologist. I know we have, like, our appendix, and there's a couple of other ones in there. It wouldn't surprise me if there's more, but I don't know if it's 180. And also, if that 180, because Tarzan, that measurement could include during development as well. So it may or may not have those other components in that measurement. Exactly, a lot. Indeed, Margot. Cat on me, excellent, NMD. I make him cite about... Oh, sigh. No, you don't. No, you don't, Smite. You stop that. Stop that, Smite. Can't unsauce my pizza once the sauce is on. Or fortuitous putting back the toothpaste into a toothpaste tube. It's very difficult to do.
At them by looking at the embryogenesis of birds and actually looking at how they develop. And he's interested in how birds actually lost their tail. He's also interested in the transformation of the arm, the hand, to the wing. He's looking for those genes as well. And I said, well, you know, if you can find those, I can just reverse them and make what I need to make for the 6th graders. And so he agreed, and so that's what we're looking into. If you look at dinosaur hands, Velociraptor has that cool-looking hand with the claws on it. Archaeopteryx, which is a bird, a primitive bird, still has that very primitive hand, but...
And so there's the pigeons. The idea, again, sonic hedgehog signaling could be the goal for that as well, is to alter that signaling pathway in this developmental stages to make something more raptor-like, as it were. Guys, go check out Lovely Freshie, another web dev, game dev extraordinaire. We love you, Freshie. You handsome devil. Gotta hand it to... Oh, Murgle. Well done, Murgo. Well done. Hey, pizza. What kind of pizza for Fortuitous Traveler?
As you can see, the pigeon or a chicken or anything else, like a bird, has kind of a weird-looking hand because the hand is a wing. But the cool thing is that if you look in the embryo, as the embryo is developing, the hand actually looks pretty much like the Archaeopteryx hand. It has the 3 fingers, the 3 digits. But a gene turns on that actually fuses those together. And so what we're looking for is that gene. We want to stop that gene from...
So again, if you're looking in development, this is an embryonic pigeon, which very much looks like our dinosaur hand, and it's the same principle that's governing it. So again, it's the genes are there. It's just some things like how Coffee Rocks was asking about the apoptosis and the cell death and the reabsorption. That's the similar function here. If you can stop that from happening, then you would maintain this kind of similar structure to it. And then the other side would also be true if we had the modern day Archaeopteryx. You want to do the opposite, right? You want to show necessary and sufficient. What you'd have to do is to show, yes, this gene is required for this, so it's necessary, but it's also sufficient. So in the Archaeopteryx, if you could make it fuse, then would you create more of a pigeon arm? Then you show both sides of things, and that would be the ideal thing. Hi, Alex Vixen. Why are bees evil? They do pollen things. Ooh. Alex Vixen. Hi, Alex. And also, Epoch says, those did a pretty great job of changing the structure for winging it. My God, y'all are both air horning it tonight. Thank you, Alex. Thank you, Epoch. How are y'all doing today, guys? Go check out the lovely, amazing Epoch as well. Maker and crafter extraordinaire. How you doing, Epoch? Welcome on in, Epoch. Penguins may act as a safe house for beneficial bacteria after dysentery or cholera to boost. Yeah, I mean, there are still astronomy show situations where it is worthwhile, but in the developed world, it usually is. It's just called vestigial, and that it also the reason to astronomy why it's probably still considered as more vestigial is you can remove it and be fine, and it does short circuit, as it will sometimes. I just got done watching Father of the Bride. Is that a scary movie? A silly movie? A goofy goober movie? As long as it was good, Epoch, and you had a nice time. Sounds a little scary.
Gene turns on that actually fuses those together. And so...
Heartwarming and silly, then good. It sounds like a Bernie Mac movie. It's not, I imagine. But it sounds... Steve Martin, okay. For some reason I was thinking Bernie Mac and...
Together, we can
Giraffe Killer! Stopping by to tell you stay away from me, and oh my God, I love you. Hi, Giraffe Killer! Guys. Giraffe Killer, folks. Giraffe Killer. Yes, you're right, Murph. But I don't know how that is relevant. Guys, Giraffe Killer is an amazing gamer extraordinaire. Please go follow Giraffe Killer. He is named... He traveled to Africa. He dressed up in a giraffe outfit, and he was... preventing poachers from killing giraffes. The man's a hero. He was in the military. That's why he's named Giraffe Killer. He was in that particular outfit, again, to trick the poachers. He now plays games on his stream, so please go follow him. We'll give him a fancy pants shout out in just a moment. He's a hero to many. He's never killed giraffes. He's being silly on that. He actually took out the poachers that were hurting giraffes. Giraffe Killer's having a lovely evening over with playing with Trey and Cricket. So he's being a little bit of he likes to be self-deprecating here. You know, guys, we don't like self-deprecation here, but poor Giraffe Killer likes to not highlight his amazing work. So guys, please go follow Giraffe Killer, a hero to so many giraffes and people. Hi, Ariella. Welcome on in, Ariella. How you doing today, Ariella? Welcome on in. Actually followed Stream Elements for a second. I was like, wow, Giraffe. There's Giraffe Killers in tardigrade. Giraffe Killer, you are breathtaking, handsome, kind, just all around beautiful soul. Don't ever believe what Trey tells you. Okay, and Ariella will get you a fan shout in just a moment as well, ma'am. Welcome on in. Who is the Ted Lunar Bear? This is Jack Horner. We're listening to. Welcome on in.
Looking for is that gene. We want to stop that gene from turning on, fusing those hands together, so we can get a chicken that hatches out with a
Giraffe Killer.
Science, know you. You're the cutest, and know that you deserve it, Juno. You a star over here. I'll reverse the card like Uno.
You're breathtaking.
All right, Giraffe Killer. Giraffe Killer, don't make me duel you. And then don't make me get the mummy after you. Yeah, Draft Killer. Don't make me have that happen. Someone who's had his appendix literally... Whoa! Has it literally exploded? I've had need for that backup gut flora twice since that. It wasn't vestigial for me. Gotcha, astronomy show. Gotcha. Yep, Ariella, this is Jack Horner. Danny worked with Jack Horner, so there is a direct connection there. Yeah. Sundry, that is a fox, yes. And Alex, we have the fox is in the band now.
Science. We like experimental
right here.
Science. We're curious, not judgmental. Always good vibes on Science on Spring. Time to learn live with Science.
The people in Jurassic Park, yes, Smikes. The dino, what is his name? The lead gentleman in Jurassic Park 1 and 3. Now, Alan Grant. Alan Grant was based on Horner.
Three-fingered hand, like the Archaeopteryx. And the same goes for the tails. Birds have, basically, rudimentary tails. And so we know that in embryo, as the animal is developing, it actually has a relatively long tail. But a gene turns on and resorbs the tail.
So they're trying to turn off that gene that reabsorbs the tail, and for some reason, it leads to embryonic lethality, which we don't know yet why that is. Guys, what I do know is please go follow Ariella Studies, another science communicating artist here on the stream, and scientist as well. She is on her affiliate push. She is very close to getting that affiliate unlocked. So please go follow the lovely and amazing Ariella Studies and help her get over that final line of that. Very, very close to getting that affiliate status. Just a few more streams and she'll be there. Amazing artist. Amazing soul. Very kind and lovely person. A set of genes causing lethality, a lot of things end up causing it. So, Sundry, I think part of it is if they're trying to turn off a singular gene, it's not cause and effect, right? You don't turn off one gene and it has one effect. Genes are a web, usually. There is a huge connection between so many factors. And if all of a sudden if one gene is turned off, it's going to be hitting a bunch of other factors as well. And that's probably where the lethality is coming on in. So it's, yes, you're turning off this one gene and that maybe isn't responsible for the lethality, but this thing that it regulates 2 steps away could be the thing that's causing it. Ariel, you're going to be getting it every time you come in here, okay, Ariel? Okay, Ariel? Okay, it's done, Ariella. Genes are like, what? Exactly, Murgle, exactly.
Gets rid of it. So that's the other gene we're looking for. We want to stop that tail from resorbing.
Draft Killer, have you heard about the pants? Draft Killer, have you heard about pants?
I like your pants around your pants.
Did you know that, Draft Killer? Did you know that about Trey? One more time for Draft Killer.
I like your pants around your pants.
So, Draft Killer, if you type exclamation point pants, you will trigger Trey singing, I like your pants around your pants.
This is the way. What we're trying to do, really, is take our chicken, modify it, and make a chickenosaurus. It's a cooler looking chicken. I mean, but it's just the very basics. So that really is what we're doing. And people always say, well, why do that? Why make this thing? What good is it? Well, that's a good question. Actually, I think it's a great way to teach kids about evolutionary biology and developmental biology and all sorts of things. And quite frankly, I think if Colonel Sanders was to be careful how he worded it, he could actually advertise an extra piece. Anyway, when our dino chicken hatches, it will be obviously the poster child, or what you might call the poster chick, for technology, entertainment, and design. Thank you.
So Draft Killer, the reason we watched that is because they came out with this paper where they were able to modify a genetic pathway called the sonic hedgehog signaling pathway, and on a chicken leg where you have scales form feathers. And it is, again, by just altering this genetic pathway called the sonic hedgehog signaling pathway. And it's and Draft, listen, Draft Killer, that's 100% a law. They named that gene, so that gene was discovered 30 years ago. It was named after the video game character Sonic. Like, the video game had already come out, and they named it after the video game character because and there's Draft Killer, there's no reason for it. There's nothing to do with speed. There's nothing to do with hedgehogs. They discovered it in fruit flies, right? There's nothing blue. There's nothing. It's just, they were like, we really like this video game. That's what we're going to call it. And now Giraffe Killer. There's 2 additional ones in humans that, like, of that genetic element that were discovered. And they are called, so their list is right above me now, desert hedgehog and Indian hedgehog. And the reason they're called that is because Sonic Hedgehog was named first, and they were like, well, these genes are the same family of genes, so they act in similar ways also in development, and they're similar enough where functionally we can call them in the same group. So we have to call them something hedgehog, because together it's called hedgehog signaling. And so there's Sonic Hedgehog, and they're like, well, I guess we'll go with Desert Hedgehog and Indian Hedgehog. I don't know why they picked those, but would have been fun to pick. Yeah, I know, right? But Ariella, the issue is it's the hedgehog signaling pathway, and then all of a sudden they went with other hedgehogs there as well. I think it was because the gene looked too spiky. Yes, no, you're not. The mutant looked spiky, Gwen. But here, we can actually Google hedgehog mutant Drosophila.
In dreams, one is not tethered by earthly limitations.
What does that mean?
Sleep well, Molasse! NMD96, sleep well, your face, NMD96. Sleep well, madame. NMD, I hope to see you tomorrow, but if not, I will see you next weekend. So, like, something like this, there's little spikes that are coming out as well. Like in panel A, there's growths coming off the embryo. So there is that similarity, but again, they were actually big fans of the video game, which is kind of neat. Used to drive me nuts when looking up stuffs. Too sciencey or wordy, completely unrelated term, confusing. I mean, fortuitous, you're not wrong. Because if you look at the title of this paper, I mean, I had to sit and read this paper, title, 4 times to figure out what they're even talking about. They could have named the tail. Yeah, that would have. I mean, Dame Karen Smikes pointed out at the beginning that if Tails named Tail, is Tails based on mutant Sonic Hedgehog signaling, and that's why it has 2 tails. Unfortunately, they were not that clever because the game came out first, but that would have been a nice connection there. Picard once said, "This is what it is to be human, to make yourself more than you are. You, Giraffe Killer, are more than any of us are. You saved animals on the tundra of Africa. You, sir, are a hero. Thank you for all you do, Giraffe Killer." Okay, so let's actually tundras or deserts, Mikes. There actually are frozen tundras, not in Africa, but just in general. And it's just a factor. Guys, let's quickly watch this video from the actual research group. So one thing they had to do, thank you, Giraffe Killer, I love your face. One thing they had to do, was they had to do embryonic injections of chickens to get this kind of effect to happen, to have them demonstrate the feathers growing on the those regions of the legs that were scaly beforehand. Giraffe Killer is a gentleman and a scholar. In fact, Epoch, let's give another shout out for this lovely Giraffe Killer, because they're a gentleman and a scholar. And just, you know what? Giraffe Killer, you know what you are? One of our...
My best friendo!
One of our best friends, Giraffe Killer, chat. Go check out Giraffe Killer, because, again, one of our... Giraffe Killer, if you type exclamation point BFF, if you type exclamation point BFF, you can get that clip as well. Being a best friend. When a PhD tells you something that you're not... I love your face, Max. Love your face. Okay, let's watch this video. This is from the research group demonstrating how they did the embryo injections. Never what, never who, what, wise man.
I like your pants around your pants.
So it's a really, really cool video because actually the folks in the lab put out this video independent of anyone else doing anything. They actually put it on. This is a corresponding video to the protocol that they put out in the paper. So in the paper, they actually had a supplemental video where they were trying to do and show off in video form what they did. So they were really making sure that anyone could do this kind of technology that they're working on. And that's, I think, very, very cool to be able to see and very noble of them to do. So they're getting the right embryonic stage. They check the eggs to find the vein in the egg because they have to, right? They have to inject it into the egg. They can't just inject it like loosey-goosey into the egg. Has to go into the vein, right? And so they identify that particular region. They create a window into the egg with a little solder, and they don't break the membrane. So I'm sure this is a very technically challenging component to do. But draft killer, listen, it does make sense, and it should make accessibility like important. But what's wild to me is there's a lot of journals draft killer where you're limited on the amount of data you can actually put into the paper, even if it's supplemental, like it's not in the full body. They limit you on how many things you can add. And then you're even text wise, like in supplemental text, I remember I would write out paragraphs for the methodology, and then it was like, you need to remove this because it's too long. Like, why? It's online, it's in a supplement, who cares? This is helpful for the next person. Like, no, you need to remove half this text. All right, whatever. There's again the little window, and now they add mineral oil to make it more visible. You want to do something great, exactly, so that anyone can do it, draft killer. That's like the goal. And I like how they made this video, and my bet is that the video wasn't allowed fully in the paper, so they just made it and put it out online and linked it. So like, here, you want to do this? Here you go. And it's cool because like there's the vein, and then here's the injection method. And actually, what's crazy, that machine that she's using is the same thing that we did for embryo injections in an ant. So the apparatus they're using is very, very similar to what we use in other organisms. Epoch, oh my goodness, Epoch, listen. There are journals that charge you money based on if your figures are color or if they're black and white. And Epoch, all those journals are online only. They're not published in paper in any kind of physical form. And yet they have different charges for color and black and white figures. So Epoch, you think you're being silly with me here. But actually, I've had to change some of my figures to black and white because we didn't want to. The difference is like $50 a figure to $500 a figure, and it's a historical thing, but yeah. And not every publisher has insane profit margins, Ariel. It depends. There's society journals. Society journals means that they use the money. They're a nonprofit. They use the money to do to put on conferences and events for scientists. And then there's the journals that are solely for profit, and those are bringing in the coin and not putting on those as special events for others. I know you know, Ariella, but not everyone might know that. I'm too Italian that this comes Italian to me. Yes. You could email, as Sundry says, the authors. You could also use a program called SciHub, which I would never advocate for because it's a workaround to let you gain access to a paper that's behind a paywall. I would never endorse it, but if you were to use it, that's one thing that you can use as well. So SciHub does it, and then you can also email the authors. But yeah, the money overhead, Smikes, is that they have some infrastructure. They have a lot of secretaries, and I believe they have full-time staff of editors, but all the reviewers, the people who review the papers, get no money. It doesn't matter if you review for Nature or if you review for the lowest of the low in terms of journal. You're not getting paid for it, ever. You might get like an email at the end of the year saying, "Thanks, friend." There is the injection into the vein. It's remarkable that they can do something like this. Right, they're going through that little membrane on the egg. Oh, and there it goes. It's in. You can see the color. And then they reseal it with a piece of... They clean it and reseal it with tape and then put it back in the incubator. And now you have a chicken that'll grow, instead of scales on its leg, it grows those feathers. Which, let me scroll down for y'all. Just to get another look. And there we go. And here's the control and there's the mutant. So again, the control... Here, this is DMSO injected, which is like nothing extra in there. And then here is the genetic treated, and there you have the feathers happening there. Draft Killer, I'm grateful that you're here. Is Scotch tape? Probably 5-star. I didn't read the methods, but I imagine it's just like Scotch tape. Tends to be university grants, which are small, only semi-directly correlate with publishing money. Well, Sundry, most of those grants aren't from the universities. They're to the university lab for the lab that's applying for it. Like from the National Science Foundation or from the NIH, which is the National Institute of Health, or Aging, or a foundation. That money is the lab that's applying for it. And then afterward, Ariel, you did not get auto-modded. There's nothing, Ariella, that you got flagged for. I'm not sure what happened, Ariel, but you did not get flagged for anything. But yeah, so that's where that money's coming from. Other chickens, fortuitous, they didn't do any social behavior on that front. No. Having a dance recital tomorrow, excellent, Epoch, excellent. I'm very curious as to what life is like for a researcher scientist.
In dreams, one is not tethered by earthly limitations.
What does that mean?
Come.
We can finish with that, Draft Killer. Draft Killer, I'm actually going tomorrow morning to New York for the day. I'm hanging out with a couple of friends. We are hanging out with Thug Shells tomorrow. She's gonna be in New York. Actually, Draft Killer, if you're in New York City, you can come meet us. We're going to Central Park. We're gonna hang out. One of my moderators, Andrew, Thuggy, Towards Night, who's another freestyle rapper, Schmakeys, who flew in from Germany, and a couple of other friendos are meeting up. So if you're in the area, everyone can meet us up. We're about to be around Central Park around noon-ish, a little after, probably 1, somewhere around there. So come hang out with us. But yeah, Draft Killer, daily research life depends on what career stage you're at. Undergrad versus grad student versus postdoc. Grad school and postdoc, I worked about 16 hours a day, probably average, most days of the week. Like, that's including weekends. And you go in, there's a lot, it depends on what kind of science you do. If you're doing computational science, it could be identifying problems in your code, and you're trying to mine data from like sequencing experiments, let's say. Like, you're mining through data to find out what the sequencing data actually means. If you're doing wet bench experiments, meaning hands-on experiments, some of those, throughout the day I would run several different experiments, and those experiments might be lasting 10 hours. It might also last a week, depending on what experiment you're doing. So some of that, is in between time you're setting up a different experiment. Hi, Kixie! Welcome in, Kixie. You might be setting up a different experiment. You might be taking care of your stocks. For me, the stocks were, in graduate school and undergrad fruit flies. So you're taking care of the animals. You're feeding them. You're making sure they're fed, that everything is going well with them. You're also, if we're doing genetic crosses to make another line of mutants, you're working on those. You're cleaning sometimes, like your lab area needs to be cleaned up. Because you have to work in a sterile environment for some experiments. Microscope time. So Draft Killer, if you want to take high-res images like this, usually it's some kind of scanning microscope. When I was taking images of fruit flies, brains, or ant brains, sometimes I would go to that microscope, and you can program it to take the image. And you can set it up to take, but what that means is, like, I know one of my ant brains not too long ago took 3 hours to image, one brain. And so, like, those are the things. And so you as a grad student or a postdoc have a project, and that project is part of the lab's mission. Whatever the lab is studying, it fits into the lab's fundamental question. And so you have a project that's yours, but it's into this overarching theme of the lab. And so that's what, you might have multiple projects as well. I did Kixie. We actually, Kixie, have ant brains that we can look at under the microscope here. Remind me to do so on Monday for Microscopy Monday. I do have ant brain samples that we can look at under the microscope. You can dissect. And so like Draft Killer dissections as well take time. On top of that, if you're in grad school, you might also have classes. You might have to teach as a teaching assistant, or you might have to do your own classwork as well. So some of those factors. Get that from my own work. A programming project takes time contributing. Yep. So and you might also, Draft Killer, work on other people's projects as well. So help out other people's projects in smaller ways where maybe you're not the lead on it, but you're doing a specific experiment that maybe you're an expert in to help out your lab mate. And Sundry, we do Microscopy Monday every Monday. We look under the microscope. We have amber that's 30 million years old with insects in it. We do pond water. We have a bunch of samples that we look at on Mondays, so make sure to tune in for that. On top of that, Draft Killer, we also do like you'll be reading papers to read about the newest data in your field to stay up to date on the science. So when you're designing your experiments and asking your questions, you're doing it the most informed way possible. That's probably the regular day-to-day. It was my experiments would take just out, and like you also would run replicates, so it's not one experiment in a like a void, but like this repeated 30 times, and it just keeps going and going and going, and it's just there's just so much on that front. So yeah, that's a lot of it goes into. There's busy work. There's also the actual experimentation component. There's a lot of troubleshooting as well. So figuring out ways that things don't work is very common. That actually probably goes not highlighted in a lot of those questions. So yeah, there's a lot of things that work on that front. But Draft Killer, if you have any specific, and paper writing too, like writing up and analyzing your data, right? So like making figures that are easily accessible to a broad range of people, what they mean, how they look. It's not easy and not straightforward, but so you have to get on that as well. Yeah. Ariella, so Neanderthal, so the auto mod is set on the higher side because we do have young kiddos who sometimes come in here, and Neanderthal, the auto mod, sometimes like flag it and be like, because apparently it's used in a derogatory term towards people, so it flags it. It's like, hey, is this okay? And then we permit it, and then it's fine to use. Sometimes other weird phrases get flagged as well. It was like, hello there. Not sure why they did. Hi, you.
Hello there.
You. All right, chat. That I think is where we're gonna round out the night.